the practical 13. I think I put the data sets, right? In the
Can. But it's still like on the way lah.
Oh, okay. Yeah, because I forgot. I totally forgot yesterday. I remember I have to to release the data set. Yeah. So that's why I know it can be take some times, but and this time the data set is not the subset. Is we need the entire data set. That's why sometimes it takes some time to upload. Okay, maybe we wait a bit before we can start. I think some of you, sent me an email regarding that it was not able to run, right? The repeat masker or something. Yeah. So I think one of the reason I think is because of the setting. Somehow the setting of your Galaxy was not permit you to run, and it was not able to recognize the defarm data set. So in that case, I think if you really cannot do it, right, you can get the result from your coursemate. Yeah, because of that. I think I remember some of you managed to do, right, the repeat masker. Repeat moderate, you maybe it will get the no output, because remember that you use the SRR data set that in the spectrum, right? Those are only subset. So the assembly generated from there was not actually a good assembly. So sometimes it will not able to detect the repeats in the repeat modeler. So that's why I say if you use a repeat masker, since we know this is Arabopsis, right? So in the model organism there, you can choose the Arabopsis thariana as your model organism when you select for repeat masker. But I don't know why the Galaxy setting issue is out of my out of what I can do. So this is a setting, and last SEM I think we're also having the same problem. So what the senior was doing that is try to register for a new account and then rerun the repeat masker. You can actually transfer the data and then run again in another new account. Hopefully that you can resolve that. Okay.
Doctor, for our lab report, for the result,
Yes.
should we just include like one best result from our team, or?
Yeah. I mean, since it's a team report, right? So you just choose the best result coming from here, from your team member, because you put it as one, you submit as one, right? Yeah. All right. Let me share the screen. Okay. So basically for you can see my screen, right? Okay. So basically for the transcriptome, right? We will need since we are not doing the de novo transcriptome, we are using a reference-based transcriptome. So what we need actually the genome file. Genome file coming, I mean in this case it's Arabopsis thariana, right? And then we also need the GFF file, because you need this file to count the number of read mapped to a particular genes. So that's why it's called feature count. features, it means like exon, intron, the CDS, mRNA, those are sequence features. So we will need that information, and it's either listed in the GFF format or in the GTF format. I think for the feature count, they will only take the GTF format. So in the spectrum itself, in the spectrum, in the in the spectrum, I upload the GFF file and also the DNA top FA, right? So those are actually for the feature count and also for the reference genome. So we can convert the GFF file to GTF file. So before we start, you see in the spectrum itself, there are about how many? Four pair of the read, right? Forward 1, forward 2, and all those read. So those are the file that we are going to use for the DE analysis. So how do you know what is this stress all about? So what you can do is you can type in the SRR number. For example, this SRR 9257060, right? You type into the NCBI. Okay, let's say you open the MCBI, right? One of this number, right? For example, I copy this number and I place it here. Then it will display, it will tell you the information. Okay, now this is a RNA-seq of Colombia, Arabocitariana, Colombia Ecotac, right? Control data set, replicate number 1, right? And it is RNA-seq. So, and then you will see that they sequence using the Illumina HiSeq 4000. Latest one, I think, is HiSeq 6050 or something, 6500. So this is 01. And you can sometimes they will show the abstract, right? To investigate a genome-wide Arabocin TE reduce, resequence the wild type transcription under control, hit and recovery, right? Conditions. So it means that now you have 3 different conditions: control, Arabocin under hit stress condition, and then after that recovery. Recovery means that recover back to the control condition. So today we only do the control and hit only. So, and here it is mentioned the construction protocol used to get the RNA-seq data. So what it is they use the I need to,
And doctor,
yes,
you said just now that we do
sorry, I lost, I can't hear you. Yeah, sorry, it was yeah.
Should we okay. Since we are doing like control under 1, under control and under hit, doesn't that mean that we would have 2 sequence? Or
We
is it
we have 2, you see, for transcriptome, right? We need replicates, right? For example, this one is replicate number 1 for control environment, right? And this one is the control number replicate number 2 for the control number 2, right? And then this one is a hit sample. So when you key in here, right, in the NCBI, it will display this. This is only one of the data set. So in order to know what is the data set that we are using, right, all the data set we are using, so you need to click on all run in here, under the study panel there, you click all run, right? So then they will list down all the SRA number, SRR number, right? And then how many byte of the data set, right? The developmental stage, how old is this plant, right? Those are 10 days and this is 13 days, right? And then when it was released in the NCBI, and then what it treatments. For example, here you have control. So first 2, 60 and 60 and 61 is a control data set, right? 62 and 63 is the hit data set, right? So from here, you can see that this is the control data set, and this one is the hit data set, right? You will need to upload all into your Galaxy, right? So once you know the data set, why is important? Because later when you do the DE analysis, right, you will need to key in the condition used for your analysis, right? So you can download from here, data set. You can either download from here, right, using the SRA toolkit, or the other way of doing that is you go to ebi.ac.uk, right, in here, then you can key in this number, and then they will tell you, okay, this is a data set here, right? You click on this, then you can download all from here, or you can download one by one, right? So when you download selected item, they will only download one, and this is a forward read and reverse read, right? They will download, and in this case, you don't need to use the SRA toolkit to convert the SRA number, SRA file into a FASTQ file. Bear in mind, in the NCBI, this file that you download, right? They download from here. Those are the SRA file. So you need to use SRA toolkits to convert the .SRA file to .FASTQ file for NCBI, right? But in the European website, European database, right? Because they share the database between Japanese, European, and China database, right? So you can directly download the FASTQ file, so you don't need to convert. So I would prefer you guys, next time if you do, right, download from here, EBI. So then you download and you upload into here, right? So for first step of transcriptome, since we are doing the reference-based transcriptome analysis, right? So what is the first step we need to do? Anyone have any idea? What is the first step we need to do?
Upload the reference.
Sorry? Upload the reference. Okay, now we upload the reference, we upload the GFF file, we upload all the data set, right? Now, so we assume that all the input is needed for transcriptome is already in your Galaxy. So what do we need to do after that? You know, the real experiment will start, right? So what is the first step of your real experiment?
Align the transcripts, the RNA transcripts to the reference genome.
Yeah, correct. So you want to align and then map the RNA-seq data, right? Short read data into a map to the reference genome. So to map, we use a aligner or mapper, what we call that HiSet tool. So when you just type here, then in the search tool for HiSet tool, right, it will appear this here, right? So in here, you want to two way of doing that. You can either choose the library in built-in genome. Let's say it's already built in here. So you see whether you can find Arabidopsis. Yeah, you can find Arabidopsis thaliana, right? But then in this case, you are not sure whether the version that upload in the Galaxy server, right, whether the GFF file is matched with this one. So better to do it is to use the genome from history. So from genome from history, we already know that this is the genome, right? So you select the genome, Oh, of course. One thing, before you do this, you need to do a fast queue, trigonometric and fast queue again, and only use the map, the pair sequence for the RNA-seq analysis, right? So for today, I just using the raw data, right? So in here, you choose the pair n, since your data is pair n, right? Then you input file number 1, file number 2, right? And then we use unstranded. Unstranded means that you don't know whether the sequence generated, right, is coming from a or sense or anti-sense orientation, right? So you use this to for our study. Yes.
Doctor, so for all the RNA sequence, we need
Yes.
to do basically what we need to do is do the quality control, is it?
Yes. You need to do that first. Yeah, because bear in mind, for any of those transcriptome analysis or genome analysis, right, quality control is very important. Unless you told the company, you tell the sequencing company say, okay, I want 2 different type of data set. One is a raw read, another one is clean read. So in that case, you don't need to perform a fast QC, because they already clean for you. They remove the poor quality one for you. They remove the adapted sequence one for you. Then you just use it as the input. You don't need to do any more fast QC, right? So it is important when you want to do a sequencing, you have to tell the company, I want the raw read and clean read. So in that case, you will get 2 different read, right? Okay.
Doctor, so for our case,
Yes.
what will be the contaminant list? Because to do the QC, you have to.
Remember last time in the genome file, genome assembly, right? You use a contaminant list and also adapter list, right? Yeah, we are going to use the same. Yeah. Right. So once you perform that QC, right, then you can actually proceed for this run, right? So for the high sets, we will use unstranded, right? Sometimes this information is important. When you want to do a sequencing, you have to tell the company whether do you want to have a stranded or non-stranded library. So stranded library is better. Why? Because stranded library, you can find out the antisense transcript of genes. But then when you use the unstranded, right, you lost this information. Yes, stranded is a bit more expensive, but nowadays I think it should be okay. Slightly more expensive only, right? So now this one we use because it's quite old data. I think it's unstranded one. So you specify the option, basically actually all other options here, whether your library is in a forward reverse orientation, read 1 is forward, read 2 is reverse, or read 1 is reverse and read 2 is forward, or both forward orientation. So by default, usually it's FR. So forward for 1, reverse for 2, right? The other thing is this we usually do not disable this no big mixed behavior, right? Cannot find any for pair. Then try to alignment in the individual map. This is not recommended, right? And also disable discordant behavior. Also, this one is actually we will choose no, right? For the summary options, there will be output for alignments in more machine friendly. You can type. So this one can be used to know your data, right? Your mapping summary here, so you can see here, right? and you can choose to print the alignment summary file to a file, right? They will print out that. So other additional options, they are, for example, you can specify the input options, skip any base from the in the input and all those others, trim any 5' end, how many base you want to trim, right? So those are you can choose, but generally we use the default value, right? Alignment options, there are many alignment options available, so you can choose different constant and all those, right? And changing the constant term coefficient, all those for the your alignment. So basically we will use the default setting, right? Scoring, we also use the default setting. Yeah, because there are many things that you can choose. This only, you only choose these other scoring options once you have an issue that's, you know, your analysis, you don't get any differentially expressed genes. Then you know that something happened to this your alignment option, then you need to choose, right? So you can also specify any reporting options, right? Outputs. So you can choose to write unaligned read into a FASTQ file. So this unaligned read sometimes is important. Why? Because some of the read, right, it may actually contain contaminants, or the read that do not fulfill the quality of the alignment option here. You will put it in the alignment read. This is important when you use a close related species reference genome. Let's say, for example, I'm not using the my RNA-seq data is coming from Arabidopsis thaliana, right? Then I'm not using Arabidopsis as my reference. I'm using Brassica napus, which is close related to Arabidopsis as the reference. Then you want to choose to write down this unaligned read to know what happened to those unaligned region, right? Yeah. So that's why you choose this. But if you use the same reference genome, you don't need to do that. And this one is you can choose yes or no also for to write the aligned file, right? So options for output, there are, I think there are two options. One is for BAM file, one is for the SAM file, if I'm not mistaken. So we use the default setting, right? Other option here usually we don't change it. So once you run, you will get a BAM file here. So BAM file, it's don't know whether you can open or hopefully you can open. It's a binary alignment file. So you will get I think you can't open it. Because they open using IGV. So then in that case, I have to change the reference genome on this one, right? So you can change the reference genome, I think here. So this one is IGV. Basically, you actually can use others IGV installed in your computer to open this file, right? So then you will get a summary file. So what the summary file show? Okay, now you see summary file show that your total pair is 100,000 read pair, right? And then among this 100,000, right, how many percent that is unmapped, aligned concurrently or discordantly, right? 0%, 0 times. So it means it did not align to the Arabidopsis genome, that the reference genome, this one here, it did not align. So it's 7.49%, right? How many align? One time. This means that it align only one per regions, right? So this is actually the one that we want. when you have aligned more than one times, right, it means that those are maybe aligned to the repeat regions, transposer elements, retro transposons or satellite. So that's why they repeat, because they present in more than one copy in the reference genome. So you will align to the different times, right? And also you have a line, this colony. So what is this colony? Is colony? There are a lot of information, in the Google. I mean, yeah. So they would so aligning. So this one they will tell you very. So for example, those align together close to each other, right? It's a concurrent pair, so they can connect to one read here. Those that align, that are far, right? It was actually having a discolon. So you can see also some other example, how they mention about what is concolony and discolony, right? I think this one, it's very clear. Align row read to the reference genome. So this one is very clear. Explain what does it mean by concolony and discolony, right? You can see, yeah. So here it explained very well, right? What is the concolony and discolony? You can check from here, right? So once you, so you need to repeat 4 times, meaning because you have 4 set of data set, right? So you need to repeat for 60, 61, 62, and also the 63, or others, right? Now we know that they are mapped and aligned, right, in the BAM file here. So the next step is we want to count the features. Count the features means that, count the feature. Count the reads, the RNA-seq read that map to a particular features, right? So then you want to, how to say, you want to know that which gene, each gene having how many read aligned to the genes, right? So then we use a tools called feature count. So in this case, feature count, they only take Arabsis Tariana GTF file. So what you can do is you can actually do a conversion. So you use the GFF read coming from a search tool here, GFF read, right? You input the GFF file, right? And then you select the, your reference genome, the one that you upload, right? And then you change it to GTF. So from GFF to GTF, then you will get a GTF file, right? Convert the GFF to GTF file. Now, since the GTF file is already built, then we want to do a feature count. So we do a count, feature count, right? Using the tools, right? Then you input the alignment file, BAM file. So let's say we take the number 1 alignment file, and the strand information must be the same that when you do the alignment. So if the alignment is unstranded, you put unstranded, right? And then you use the GTF file in here, converted, right? And the features that we want to count is we count the exon, right? Not only CDS, we count the exon. So it means in this case, you also include the 5' UTR and 3' UTR region, right? And then how we know that this, for example, those exon belong to what gene? So we need a GFF gene identifier. So in this case, you will use a gene ID. So where is this gene ID coming from? So now when you open your GTF file, right? So you see they only count the exon. So any read that map to exon, they only count. Any read that map to CDS, they don't count. Right? So they only count the exon. And then how do I know these exons, right? Those exon here. I mean, these 6 exon belong to what gene? Then it refer to the gene ID here. You can see the gene ID, right? So that's why when you run the feature count, you need to specify the gene ID. This one, it must be identical, to your GTF file. So if you put something like this, right, you won't be able to recognize, because in your GTF file is lowercase. So you must follow the lowercase. That's why you need to check your GTF file properly before you start here, right? So then you want to perform the mapping. So the format for output is gene ID, which is this one, right? The plus and then a tab, and then followed by the read count. So when you do that, you will get a count table like this, count matrix, feature count matrix. So you see, for gene number 1, right, you have an ID of 1, only 1 count, 1 read a line, right? 2, and this one also 1 read a line. So one of the do the counting. Wait, I want to show you one of the important things when you do the feature count. So there are 2 ways of doing the count, right? One is, since this is pair n, you can count them as 1 fragment. So you mean forward and reverse, align to the same exon, you count them as 1 fragment, right? Or you can do them as a individual fragment, right? So in this result here, since we choose to count them as 1 fragment, right? So when you have 1 in here, it means there's actually 2 copies of the RNA-seq read aligned to these genes, right? So you can see some is having a 1 count, and some having 0, no map, right? The genes did not express, right? Some having a very high one, quite high number. 227 is considered okay. So you can see the variable, value, right? And you need to perform and you also have this summary. So in the summary file, they will tell you. So this is the summary file, right? How many read, how many fragments. So you have to time 2. How many fragments assigned to the features? It means how many fragments of your RNA-seq data, right, aligned to the exon features, right? How many of them is unmapped, right? How many of them is having a chimera read, right? And also how many not assigned, no features. So this is a summary that you have. So basically you want to see a good number here. Actually, if possible, reduce the number in all others, right? So now you perform a feature count of 4, right? 4 set data set. Now we have the feature count now. The count, then we want to perform a differentially expressed RNA-seq, right? Since we count features, all the features and also the data, right? Then we want to perform a DE-seq, differentially express a sequence analysis. So we use a DE-seq tool. you just type here DE-seq2. So you can use a DE-seq2, right, to run the DE analysis. So DE analysis in here, how you want to perform, you use this one, data set per level, right? So now you can simply put there are two different factors. So one factor number one and what is the factor level, right? So factor level, you can have two types. One is a treated level, one is a control level. So treated level is a heat stress, right? So we put it as a treated level, and the type of the stress is what we call the heat stress, right? So you then select the feature count of the heat level, right? And so you need to put 2, and then this is a feature count for the control level, right? Then yes, your file have a header, right? So in this, then they will remove, do not count the header column, right? So count data is what, they are, what kind of count data is height, HTC feature counts and string tau. So they are, or is a TPM value, right? So because you already use the count data, so they will do the count data here, right? So for estimation of the size factor, right? So we usually use the default setting, right? There are 3 different types, either using the ratio, post count, or iterate, right? So the ratio one is used for the DE-seq2, right? And there are some others. We don't use any section for this one, right? And then any pre-filtering you enable, so they filter those low count genes, to filter out those that having the only, let's say for example, only 1 count or 2 count, right? So pre-filter value usually is 10, 10 fragment. Anything below than 10 you will consider as noise, right? Then options output, you will generate a plot, visualize for the result, sample size, normalized count. So because normalized count, we need a normalized count, right, for the plotting of heat map. So we will choose this here, right? And then so once you run, you will see the data like this. Now, gene ID, base means, right, log FC. This is an important one because this tells us whether the gene is up regulated or down regulated. And then also you have the P adjust value. And this one tells us whether your log FC value is significant or any non-significant. So for log FC, right, we usually choose 1. Log FC must be more than 1 and less than -1. Only consider as a significance, DE, differentially expressed. And the P adjust value must be smaller than 0.05. This is a lenient setting. More stringent setting is you use the log FC of 2, more than 2 and less than -2. And P adjust value of 0.01. Those are more stringent setting. But for standard, we use 1. So anything that more than 1, right? So example, this is 5. So this one, it's log FC of 5. So 5, it means that they have the expression level, in the treatment, in the heat stress sample, right? The expression is 2 times power of 5 times more expression than the control. So because you're using log 2, log 2, so let me check. So let me open the file. When you have log 2 FC, let's say of 5 point something, right? So this one equal to 2 5.13. Yeah, so you have this copy more. So when you put in calculator, how can I see? Okay, how to do it? Mathematics, scientific. So you have 2, oh sorry, of 5.13. So this one equals to 35 times, 35-fold higher than the control, right? So in the one of in this gene, right? There are 35 times more copy of RNA in the treated sample control to the compared to the control sample. So when you see something minus, right? So it's reverse. So you have 1.5 times, 2 power of 1.55 times more expression in the control sample compared to the treated sample, right? That's why when you do the DE-seq analysis, right, it's important for you to, put the treated sample first compared to the control. So if you put a control sample here, right, so then it's control versus treated, right? Then the result generated in the log FC here, it will be opposite, right? So it's important for you to know the arrangement of your samples, right? So DE-seq2, I think they also input the plots here. It will generate a plot. I think it's got heat map. Oh, sorry. Maybe it got the MA plot and the volcano plot. Let's see. Takes some time to open. Right. So you see one of them is a PCA plot, right? So PCA plot, you will see when if two of the data set, right, they are having a similar profile, they'll be grouped together. So now you can see the control, right, two control, they group together, right? But two of the treated file, they are separate from each other. So this is actually not very good. You want to see them either this one coming to here to join the 62, right? Or this up. So then it means that in this case, in the experiment that is done by that group, right? The heat stress condition was not, heat stress experiment was not conducted properly. So the expression profile for two replicate, right? They are far away. So this is actually not very good. So if this happened in your study, right, what are you going to do? You need to include more samples, right? More replicate. So maybe you're adding another sample here, sample of control and treatment, right? Then if they group together in here, right, then they actually better, right? So for control, because it's control condition, right? So you see their expression profile is very similar, right? And then also sample to this. This is sample to sample distance. So it's the correlations. Sorry, not correlation. Correlation. When you compare sample to see whether your replicate is actually grouped together or not. So although in the PCA you see two treated sample, they're far away, right? But when they put it into the coefficient plots here, so you see that it's still the control sample still form a branch, treated sample form another branch, and so it means that that's still okay. Or those they are spread quite apart right here, and the dispersion estimations, right? Dispersion of all data estimations. So you see the gene estimate, right? The one that estimate based on the data, and the fit one is your data. So they're still fitting the code fitting, right? And the final. So it means the expression dispersion is still okay, right? Then you have a histogram of p-value of heat stress. Then you see the p-value, right, ranging from 0 to 1, right? So there are a lot of them not significant here, right? So the one that's significant usually is here. And then you plot they will plot the MA plot, right? MA plot telling us, one of the dot represent one gene, one single dot here. So those that above the log FC of 2, right, those dot that with the blue color here is upregulated genes. Those that below, it's the downregulated genes, right? So from here we can know that we will be able to know which genes. I mean, sorry, not from here. From the data set here, we will be able to know which one is up and which one is down, right? And this also produce a size factor. So size factor used for the comparison, right? For estimation of your analysis, right? The normalization, this is normalization size factor, right? And also they will generate a normalized count in here. So from the normalized count, right, then you need to use this normalized count to so this, I think this one is if I know mistaken, this one is FPKM, yeah, generated normalized count, right? Fragment per kilo, yeah. So you use this one for to build a heat map of your expressions. So in here you see you build a heat map based on the genes that is coming from this normalized count. So you can see the profile here. So control sample and treated sample, right? So you see the expression profile gradient here, right? Low up to go high. So you see that this block of the genes, right, they are actually having a different expression profile, more lower in the control and a bit higher, more higher in the treated sample, right? And from the dendrogram on the left here, you can split them to become different cluster. For example, you can split from here. This belong to one cluster, and this one here coming from this, right, is another cluster, and this is the third cluster. So This clustering allow you to identify the genes that share similar patterns within the conditions, right? Within either treated or the stress condition, right? So the DE-seq, they only plot the MA plot, but usually when you want to analyze the analysis, right, you want to summarize them, you put them in the Volcano plots. So it will generate a Volcano plot. So you generate a volcano plot and you can label the volcano plot, the genes that is actually having differentially expressed here. Right, down is down regulated, up is up regulated, and the non-DE is non-significant. So you can see that majority of the sequence, they are actually not differentially expressed. Those that label, those are the differentially expressed. So this is a volcano plot that you're going to be able to generate. Right, to generate a heat plot, you you use a heat map tool. Right, from here, you put the feature count data, right? Then you plot the title, heat map or something, right? And then you use the data transformation log2 value plus 1 from my data, right? Do not compute the G-score, do not skew my data or any clustering, right? Enable data clustering, yes, you want to cluster them, right? And then cluster the row and column, right? This the method, you use this one. There are many, but generally for DC we use this one. Clustering we use the what D2. I mean there are other method. You can use average what D sometimes, but we usually use this one. Right, and then you want to label the column and the row, right? So you can use different color. Now I use red, yellow, and green, right? So you can choose, you use other color here. And then output as a PNG or PDF. So once you click, right, they will generate a heat map like this. This will be used for your report, right? For volcano plots, you use the volcano plots in the search tool here, right? You just need to input the data from DE-seq to data output, which is this one. I'm sorry, result file, right? So then the file has a header. Yeah, you can either choose auto detect or yes, you use auto detect. So you need to know which column is corresponding to FDR or PHS value, P value, log FC, and label. So how can we know? So we can now know from here. So column number 1 is the gene ID. Column number 3 is log FC. Column number 6 is P value, and number 7 is P adjust value, right? So you need to put this information in here. Okay. No, not here. This one. Right. 7631. Significant level. Right. Log FC of PHS value of less than 0.05. Log FC threshold 1. More than 1 and less than -1. Right. You want to label the significant one. Right. Label only 10 gene. Up top most significant genes. Right. And then you have other. So you either use you can label the exits here also, right? Those I can choose. And label for legends, you can usually use down, not significant, and up, right? Output options will be here. So maximum value, right? And minimum value for the So let's see, because when you plot, right, you can choose to plot the p-value minus 10 of p-value, right? So, or p-adjust value minus log 10. So let's see, try the calculator. Log, oh, sorry. So log, okay, log, no. The log value, log 2 value, FPR. So 0.05. So it's 1.3. You need to put 1.3 in the here. So Y is 1.3. And this one, it's the label for X axis is 1. Yeah. Oh, sorry. X axis 1. And this is minus 1. This one is 1.3. So when you run, it will plot the plot, right? And showing also the how to say, I think they will plot a dash line in here. So they will show a dash line here, right? And also dash line here. I think so. So you can check. Oh, okay. Have to wait for them to run. Right. So this is basically what we are doing for the DE analysis. Right. So you will generate a volcano plot, a heat map plot, and also the PCA plot. This PCA plot. And this one, sample correlation plot. So for your report, right, I would need you guys to input, this one will be figure number 1, figure number 2, right? And then figure number 3 is this one, heat map, and figure number 4 is this, right? So I need this in your report. This is for the identification of the differentially expressed genes, right? So let's see. It's still not running. How come? Oh. One, 7, 6. Okay. This is a new data set. Not all its data are available. Yeah, but let me check whether there's something wrong. Feature counts all here. Funny. 108.111. 108.111. 114. 173. Right. Okay. X. Significant tops of level. Data set available. Okay. Ayo. Funny. How come it's not generated the plot? Not sure it is the Galaxy setting. Let me try another one and see. Let's see, for example, I try another. So then I can run the volcano tops. Auto detects. Okay. Significance. Minimum number -1, 1, 1.3. So we run. Oh, I guess maybe I have to wait to generate it. Right. Basically, you get this type of result for your differential express, right? Then you can count the number of gene that is differentially expressed. So, for example, you need to this file, you save the file, right? Let's say you save as tabular here. Wait for the file to download. So you drag the file into Excel. So you see that here the header is missing, right? So you need to insert a new one and then view the file here and you copy this header. You here, copy. Yeah, so now you have the gene ID. So then we know how many gene is differentially expressed, right? You want to know how many gene that's differentially expressed. So what you need to do is you still need to do a filtering because in there they put everything, right? So you want to know how many is differentially expressed. So first thing first, we filter according to the p-adjust value. Anything that below bigger than 0.05. So for example, now anything bigger than 0.05, right? There onward is delete. Right now we know that there are 185 genes that is differentially expressed. Hence, we don't care about the log FC first, right? So there are 186. So, but another criteria, filtering criteria, is filtered by the log FC, right? So then you need to do a data, sorts the data, right? Then using the log FC from smaller to biggest. Now you see that those are down regulated, right? So anything that's bigger than -1, we don't want, right? Anything that big smaller than 1, then we also don't want. So this is the, although this one is differentially expressed, but this do not fulfill the threshold of the differentially expressed, the expression level. So we delete this one, right? So then you have this profile. So now we know that number of genes that is differentially expressed, it's 164, because -1 from the header, right? So 164. How many gene that is down regulated? So 55 gene is down regulated, and then you need to delete the up regulated. I mean, delete the down regulated from the 164, then you get the up regulated genes, right? So the up regulated ones, it's 164 minus 55 genes. So 55. So, and then you have 109 up. Down it's 55 genes, right? And this one you can write in your result report, right? So from here, we got this value. Now we know that those genes are up and down regulated, right? And we want to know the function of those genes. Then you will need to prepare a DEDs, right? So just only the ID. So what you do is you take this one, right? So this genes, all replace genes, replace all. Now you get the ID, right? The gene ID. So you can know what is these genes. Simple. Just go to let's say NCBI, or just type NCBI actually. And you just type genes on nucleotide sequence. Oh, it's a bit slow. NCBI issue. Okay. So this is the genes. Defensin-like protein. So what is the function of defensin-like protein? in heat stress. So stress response peptide, right, interact with the heat shock response. So they help to mitigate the osteotoxicity, right? As is shown in here. So defensing in humans, right? Cysteine rich antimicrobial under heat, frequently modified, and so balancing the heat stress and disease resistance. So there are actually some of the genes already well studied. So you can actually know that, just copy if you want to know the function of the genes, right? The other way of that to know, it's we use to predict the functions. So once you do the DE file, you upload the DE file here, right? Then you use a G profiler. G profiler. Yeah. So you use a G profiler to find the you put the DE gene, right? The organism, common organism, you just put Arabidopsis thaliana, select Arabidopsis thaliana, right? And then you only select those significants, right? So significant, it means the threshold 0.05, right? The enrichment threshold of 0.05. And you only show the annotated genes, right? So those are, you want to know the result of the biological process, molecular cellular components, right? And also the goal term, and also you want the KEGG and others. Once you run, you will generate this file, right? Then they tell you that the genes corresponding to what in your data, right? So those are the terms. So they respond to a heat stimulant, respond to antibiotic stress, abiotic stress, right? If so, the gene is also involved in the photosynthesis. So those are the one that is the functions predicts, right? Wait now. Yeah, those are the significant one. They show only the significant one, right? And I think they also have a plot generated. It's sometimes. Yeah, I'm so slow. Yeah, so you can see that those are the goal enriched plot for MF, for CC, and for BP, and also for the pathway they involve. So involved in the photosynthesis pathway, photosynthesis, right? Protein processing in the endoplasmic reticulum, right? Carbon fixation in photosynthesis organism, right? You have the metabolite pathway, glycosidate and decarboxylate metabolism. So those are the secondary metabolite pathway that involve in the heat stress, right? Carbon metabolism. Yeah. So you have all those information. So this figure, you need this figure for your result, right? So basically, you will need to do the go in KGG using the G profiler, put the DE, common organism, right? You choose only the significant one. Right. Use threshold of the value of significance less than 0.05. Right. And then you can choose the background. So what is the background then? Background file, it's the tabular background file. Wait, I think this background file, it's the all the genes in here, all the gene ID. So this will be your background ID. So, and then, yeah, data source. Okay. Tool setting. So those are, I mean, you just need to, input the DE file, right? Those are the significant one, right? And then uh, run using the default setting. So you choose the significant, then you should be able to get the plot file. E gene common, right? And all the, so you want everything for analysis, right? Then you exploit the plots. When you choose exploit plot, right, you will get this plot result. So that this will be used for you, to know which one is actually the one that is involved in here. Of course, you can know this from the data file itself, right? You can know from here. So because the top 10 most enriched one is this one. Yeah. So those are the considered the most significance, right? Yeah. So in here also you got Go BP, also Go for CC. So top CC is envelope, organelle, thylakoid, right? Plastic. So those are for cytoplasm. Sorry, for photosynthesis and use cytoplasm, and also for photosynthesis. So it seems like photosynthesis is involved in the heat stress. And you also can have predict the molecular function of those differentially expressed genes. So they do have the specific binding domain, right? Proton membrane. I think this one is involved in for the photosynthesis. Chlorophyll binding, again photosynthesis. Energy one, so energy, so relate to photosynthesis, right? And then you can look for the KGG. KGG, you have the first most enriched one, it's the photosynthesis, right? The second one is protein processing, then followed by all others. Yeah. So those summary they will choose to display in this plot. Yeah. Then you will need this plot for your figure. So this is figure number 5 for your the go in KGG of the differentially expressed genes, right? So I think you should be able to, let me try whether I can put the um, background genes in there or not. So one. Save. Let me upload this. Then I want to rerun this one. And then, okay. Wait for this background file to be generated. Then I can choose to input the background file to see whether it can do the enrichment analysis. So the other one is this one. Oh, okay. So just now I tried to rerun the heat map. Sorry, heat map, volcano plot to see whether is there anything difference. Hmm. Oh, sorry, my internet is a bit slow. Yeah, what happened? I also upload the manual thing. Yeah, the manual. So manual for the today's practical. How to use a volcano plot heat map. Yeah, but I did not include the goal. I mean the goal for profiler, right? So the goal profiler you can actually run by what I did today. So let's see. Hmm. Oh, I thought usually they will draw the maximum of my mind. Wait a minute. Use auto detects significance of options. Set to this a label for the point box. Label for Mhm. So what is the difference then? Oh, okay. So they will put a reference here. I do this one. Mm, it's the same. Somehow it didn't draw the line. I want to draw the line between, let's say one and also this, but somehow it's not allow me to draw the volcano plot. Let me, let me see whether we need another volcano plots. Or there's only one. So I try to use the latest version of volcano plot and see. FDR 7P631. This one do significant log ST to color 1 significance. So top 10 most significance. Top level. That's one. Mm, 1, 3. All right. I rerun again the volcano plot using the new version of volcano plot. Let's see whether later you will show us the different result. So back to this one. Now the background is here, and we can try to run again to see whether can we include the background file. Take a look. Oh, let's see whether we can change it. So basically, this is what the standard, standard pipeline for the DE-seq. So what we need, if we want to do a reference-based assembly, we need a reference genome, right, in FASTA format. Then we will need the gene annotation file in GFF or PDF format, right? Of course, then we need the read file, the RNA seq read files, right? Then once you have that, first step is you want to map the read files, reads to reference genome. So in our case, we are using the HiSet2, one of the aligner, right? Once you finish all the mapping, right? Second thing is you want to count the sequence features. So in this case, we count the sequence features of exons, right? Using the feature count. Once you have the count matrix, this will generate the count matrix. Right. Then you want to perform differential expression analysis using DE-seq tool. Right. So once you obtain the DE-seq output, right, then you would like to do you want to plot the DE outputs, right, using heat map. This to see the expression profile of those DE genes, right? And also using the volcano plots, right, to summarize the DE gene expression profile. So now you know the DE genes, right? Then extract the DE genes ID, then use G profiler to predict the functions of those DE genes. So basically, this step is to functionally characterize the DE genes to know which Go and KEGG way they are involved. And also to know um, which pathway is PE gene regulates. Yeah. So basically, this is what we want for today's practicals, right? This for the that this is a standard pipeline for differentially expressed gene analysis. So now let's see whether you will generate a better form or not. So slow. Still the same. So I believe there's nothing different. So let's see what is the result generated. Still response to temperature stimulant photosynthesis, light harvesting, right? Respond to stress. So you're still having this the same. So basically, you can include the background genes if you want to do a enrichment analysis. So this second one is the enrichment analysis, right? You see which pathway is enriched. Right. Okay. So this is basically what we do for today practical. Right. So any one of you already uploads the data sets or still uploading?
Me, but I'm still doing the wait, what did I do?
I said
I'm doing the I already done the feature count, but only for one gene.
only for one data set, you mean?
Uh yes.
But did you manage to do the feature count?
Yes.
I said okay.
I said okay, but actually the original sequence I didn't do quality check first lah. I immediately
Just
do.
run. Yeah. I mean,
Uh,
at least you get a hand on how to do it, right? How about others? Anyone manage to upload and try?
No, doctor. Actually, from my side, Galaxy today very lag. I upload 12 MB also need like 15 minutes.
Yeah. I sometimes Galaxy, I don't know. It's yeah. Maybe sometimes too many people was using. Also, they will have lagging actually. Yeah.
by the way, I just want to mention lah, if anybody is having problem with the feature count
Mm.
or the I forgot what the name of it. It's basically like the gene identifier. For the file that we use, the gene identifier is name.
It's
A name. If you use gene ID, cannot.
huh? Then you need to.
Uh-uh.
Can you show me your GTF file?
Wait, let me
Yeah. You share
open
the
this
screen.
wallet.
Yeah. Yeah. You share a screen. I stop sharing here.
Bising lah nanti.
Hey, why you do one history one by one? You can do actually all in one, right? In one history. So you say the feature count having an issue, but I see a feature count okay or green. No?
No, no. I was just saying, like earlier when I do the gene ID. Wait, sorry. If okay, let's say if I do here,
Uh-huh.
this is the file that we get.
Yeah.
Okay, first of all, the file need to convert to GTF file because
Yeah,
like
yeah, 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 yeah.
not
Yeah,
yet. But then
I
this one, if you use gene ID, then it will result in an error. Wait, let me run it.
yeah, because you are running using the GFF file.
No, but I already did correct it. But let's say if I this file wait I open it again. This file
Yeah, this is GFF file, right?
no, I already converted to GTF.
Yeah, but how do you convert? Can you show me how you convert?
I just go here lah.
No lah, you cannot do that.
Oh, cannot sorry, wait, where is
You cannot do that. You cannot do that. You have to use a GFF finder. GFF, you have to use a GFF read I mentioned just now to convert.
So but is my result okay? Wait, how do I show summary is it? Is this the file to see if it's okay? Wait. Preview. Is this okay or
It looks okay, but you see now the features that you count in there, right? So although you count the feature assigned already, but then you don't know whether the exon is linked to the gene or not. So for this one it's successful. Can you use a refresh button? I want to see what you did. I mean the refresh button for the for your feature count, the one that you want to restart that run. Yeah, run the job again and go down. So okay. So where is the you cannot use done. If you use name, right? So can you open your you use the GFF gene identifier, you use the name, right? Can you open your GFF file, the GTF file? Yeah. The I want to see the yeah. Okay. You will notice one thing. They maybe have names in the transcript, name in the gene and name. So anything that they found a name, right? They will classify them those exon below to that. Then that is wrong. Somehow quite slow. You go to preview and see. Oh. Reload again. Yeah. Refresh again and see.
So what should I use as identifier in this case?
You
Is it-
cannot use the GFF. If you use the GFF, right, it cannot
Oh,
recognize the identifier. So because your there will be a lot of names, you know. So yeah, so then it will causing the issue. So what you usually do is we use the ID. You can use ID rather than a gene. But then I think the best way is to do is convert to GFF GTF file. So in this case, right, you see gene under the gene, right, you have ID, right, and then the name, and you see the name have different. No, sometimes you have a name of AT1G, and sometimes you have the name of ESR1 something, right, direct, right. So that might be confusing your the feature count.
okay. So I'll
Yeah.
try using the converter first.
Yeah, you
Wait,
use a GFF converter. You just need to use a GFF to convert to GTF only. So you use a GFF read. Okay, I show maybe I show you now the how you run. Okay, let me share the screen. So to convert the GTF file, right, to GFF file, you use the GFF read. So in here, GFF read, right? Use this one. So you input your GTF file, GFF file, right? And then you choose the reference genome from history, which is the one that you upload, right? And then you want to convert the features to GTF. Then you just run. So once you finish, you will get a GTF file like this. So they will tell you which one is exon, right? And this exon is linked to which ID, gene ID. Right, you can see the gene ID. So this is basically what we want. So they will only count the features, and these features are belong to one genes, which is this one, right? And then they will count another features. They found another exon here. So those exons, it belong to gene number 2, right? So this is why you need to have the identifier as a gene ID. So in your case, maybe you can use ID as a gene identifier, but I think it's have to check properly because the ID, because later we are going to use ID, right? I mean the ID for DE analysis and all those. Sorry, not DE analysis, for the Go Profiler and others. So I think it's better for you to choose properly, right? So in here, you should use ID, not the name. Yeah. But you see, ID, there will be one issue. You have also ID for chromosome, right? So that will affect your result. So I think better for you to convert to GTF file before you run. Okay. All right, then anyone having what try to run? Anyone manage to run it? Only one, only Emma. Others, but can you guys follow? Maybe you can try to run it first. If you have any issue or something, then because tomorrow morning we have our last practical, right?
Doctor, actually I have a problem about the genome practical.
What is the issue?
Because I did the genome scope,
Ah.
I did the repeat masking
Uh-huh.
using the reference genome in Spectrum, and then the result is the whole genome 99% is repeat.
How is possible?
I also don't know. That's why I shocked. So I didn't continue after that.
Can you show me?
No, not now, doctor, because I don't know why I cannot log in into Galaxy Europe now.
Oh, okay. So just now also I'm having an issue to log in Galaxy Euro. I don't know why. Sometimes they are not stable. Maybe tomorrow or tomorrow morning you remind me, then you show me.
Okay, doctor.
All right then. Yeah, then that's all for our class today. And make sure that you take the attendance. Right, especially for those international students. All right then. And see you guys. All
Thank you,
right.
Doctor.
Thank you.